Are also very lucky to have Dr. Danielle Dick here with us, professor of psychiatry and the inaugural director of the Rutgers Addiction Research Center. I think some RARC members are here, which is great. Hope you'll tell us a little about the center. So very much looking forward to hearing about your talk about advances in genetics to prevent substance use disorders. So thank you so much, Danielle, for being here.
Absolutely. Well, thank you all for inviting me and for tolerating my kind of crazy schedule here today. So let me share my screen and get started. Okay,
Looks
does
good.
this look good in the right format for you all?
Yes, thank you.
Wonderful. So, um, as Carolyn was saying, I'm Danielle Dick. It's lovely to virtually get to be here with you all. And I joined Rutgers about 4 years ago now to build and direct the Rutgers Addiction Research Center, which is actually an umbrella center with the goal of bringing together researchers from across all the Rutgers entities who are doing addiction-related work and then connecting the researchers to our clinical enterprises as well. And so if you are interested in learning more, I'll have a slide at the end, but you can go to addiction.rutgers.edu. We have a listserv. We kind of collate everything that is addiction related going on every week and send out information. And everything that we do is open to anyone. So we would love to have you all join. So, what I'm going to talk about today is really the work that's ongoing in my lab. And so, I am trained across genetics and psychology, and my work really spans basic science all the way through to translating that knowledge into improvements in human health. I know you all are largely at the Cancer Center, and it's interesting in the sense that in talking with several of my cancer colleagues, alcohol or substance use is often kind of a risk factor when you're thinking about cancer outcomes. And we think about it as an outcome in and of itself. And so what I'll be talking about today is influences on use of substances and the development of substance use problems. But of course, that also has downstream effects for all of these other biomedical conditions that are related to substance use as well, too. So if we can prevent risky substance use and problems, we can also reduce the likelihood of all these
I
other
want more food
downstream
too, but I'm
effects.
so full, I can't eat anymore.
So, thank you for that. So I'm going to start with just a few disclosures. I wrote a book for which I received some royalties from Penguin Random House. I have spun off a startup company, which is actually going to be in the innovation portal here. I'm in the Rutgers Health, the new building today, and I'm on the advisory board of a couple other companies. And I mention this because these are things I never imagined myself doing. I'm an academic through and through, but these are really initiatives I got involved in an effort to kind of spread science beyond the walls of academia. And I'll actually talk a little bit about that today and how I ended up doing some of these things. So I am going to start with why. I like to start my talks with this question of why. And if we were all together, I would kind of ask by show of hands. So maybe if folks are willing, if anyone has read this book, kind of, either drop it in the chat if you have read this book. I will say I have asked this of thousands of individuals in talks that I have given, and I have only ever had a couple hands go up. And this is actually a best-selling book. Millions of copies have been sold, but it's a business book. Oh, I see somebody out there has read it. And I think that's one of the reasons why, because it's a business book, a lot of academics haven't read it, but I think it's very relevant to our world and what we do as well. So in this book, essentially, Simon Sinek argues that most companies focus on what they do. So Hewlett Packard makes printers and they focus on doing it really well. So they try and make the very best printers. But great companies, he says, focus on why they do what they do. And he gives the example of Apple, whose motto wasn't, we're going to make really great home computers. It was, we create technology that will change your life. And that allowed them to go seamlessly from home computers to iPods to iPads to iPhones to technology that literally has changed our lives. And I think the parallel here is that as academics, we very often become entrenched in what we do. It's what we say to each other at conferences. Oh, what do you do? What do you do? But when we stop to really think about why we're doing what we do, I think it can drive our work in really interesting ways. And so, with that by way of background, I'm going to start with why I work in the area of substance use disorders, which is that substance use is the leading cause of preventable death worldwide. So there are 11 million lives lost globally. And, we throw around a lot of numbers as academics. But what that translates into is more than 60 jumbo jets going down every single day. So, in the time that we will be together today, more than 2 planes will have crashed. And I think that when we really think about the magnitude of how many people are affected personally and by rippling through the families, the neighborhoods, the communities that are affected, it's important for us to think about that, why we're doing what we do. So my work focuses on kind of this big question of why are some people more at risk? What causes substance use disorders? Substance use and substance use problems, tackling them is really a grand challenge because most people choose to use alcohol or other drugs recreationally at some point. In the United States, it's about 85%. But only a percentage will go on to develop problems or to use in risky ways that really impact their health. And so we know that part of the reason why some people are more at risk than others is found in their genes, and the other part of why people differ is found in their environments. And so the heritability, or how much of the differences between people is attributable to differences in their DNA, is about 50%. So about half of the differences between all of us and how much we say use alcohol or like alcohol can be found in differences in the DNA we were born with, and the other half is differences in our environments. But we also know that there is no gene for substance use disorder. There's no gene for alcohol problems or other drug problems or depression or most of the complex outcomes that many of us focus on. Instead, what we know are there are many, probably thousands of genes of small effect that all come together to influence why some individuals are more at risk than others. And so the way that we help people conceptualize this, with full credit to one of my dear friends and colleagues, Jane in Austin, who's really the world's leading psychiatric genetic counselor, is that we all have a mental health jar. So if you imagine, those clear glass mason jars, and when that jar is full, that's when someone is actively experiencing problems. So that's when they might meet criteria for a substance use disorder or for depression or anxiety. And so what are the things that can contribute to that jar being full? Well, there's genetic factors. So we all are born with some marbles in our jar, and it varies as to how full that jar is when we start life. And then the other thing that can go in that jar to lead to it becoming full is environmental factors. And so we know that everyone has some vulnerability to substance use disorders. We all have some marbles in our jar, and that risk is really distributed like a bell curve. So some of us are lucky and we started life with just a few marbles in the bottom of our jar. Others are unlucky and they might have jars that are 3/4 of the way full. And lots of us fall somewhere in the middle. And then, of course, what happens is that over time we might accumulate environmental risk factors. And so you can see how you might start life at lower genetic risk, but accumulate a lot of environmental risk, and that can lead to one's jar being full. Another person might start life with a lot of genetic risk And then it doesn't take as many environmental risk factors for that jar to become full. But key things that I think this also emphasizes, no one is born destined to develop problems. No one's born with a full jar. And we can essentially also think about how we can remove environmental factors and add protective factors so that the jar is taller before it gets full when we think about prevention and recovery. And of course, we all have our own unique jar. We have our own set of genetic and environmental factors. And this is really where basic science intersects with this broader conceptualization of knowing what causes substance use disorders. And this is the area where my lab works in trying to identify the specific genes and environmental factors that are leading to risk so that we can ultimately develop more effective and tailored prevention, intervention, and treatment. And so, what I'm going to talk about today is really new advances in substance use genetics. And then I'm going to spend the latter part of our time together talking about how we're trying to translate those new advances into practice. So, what do we know, just so we kind of can start on the same page, about gene identification projects for, in this case, alcohol use disorders, because that's the furthest along, the biggest sample sizes are available. So to orient you to this figure, what this is showing is every dot is a GWAS, a genome-wide association study that's been done. The green dots are studies of alcohol consumption, how much people are drinking, and the red dots are studies of alcohol problems. So individuals who are actually experiencing problems associated with their substance use. And so what you can kind of see if you squint here is that over time on the x-axis, you see the samples that are being used for gene identification are getting larger, such that the largest gene identification study for alcohol use problems came out a few years ago, was a little over 1 million individuals. And then this is how many genetic loci had been identified as increasing risk associated with alcohol use disorder. So as our sample sizes have gotten bigger, we've identified more genes. And then you see this one outlier over here. This is a study of alcohol consumption conducted in about 3 million individuals, which has identified a number of several hundred genes. But the problem is that we know that the genes that influence consumption are not identical to the ones that influence problems. And we're probably much more interested in the development of problems and using in risky ways than the individual who's having a glass of wine, for example, with dinner, who might be drinking frequently, but might never go on to develop problems. So while that's an important outcome, it's clearly not going to get us all the way to understanding the development of alcohol use disorder. Now, my background is originally in twin studies, and while that might seem, like a quaint study design now that we can actually measure the genome, there were decades of twin studies that essentially taught us a lot about how genes influence risk for substance use outcomes. So we couldn't identify the specific genes, but by comparing identical and fraternal twins who shared all or about half of their genetic variation, we could learn something about how those genes were operating. And what we learned is that alcohol use disorder, other substance use disorders are frequently comorbid with other behaviors related to impulsive action. So childhood conduct problems, adult antisocial behavior, personality traits related to impulsivity. And all of these different outcomes, which are frequently seen clinically as hanging together, they all appear to be influenced by the same set of genetic influences, meaning what we learned is that there is a latent factor, a shared set of genetic loci that are impacting all of these outcomes. And this underlying genetic predisposition toward impulsive action, or sometimes called behavioral under control, is highly heritable, more heritable than any of the individual disorders themselves, which can be different manifestations of this underlying predisposition toward impulsive action. And so, if you're interested, for example, in the genetics of alcohol use disorder, what we know is that 2/3 or more of the genetic influences on alcohol use disorder and other substance use disorders appear to be operating through this shared factor that's influencing all kinds of impulsive outcomes. And so this really, led me to think, well, why are we studying these different DSM disorders as if they're separate things when there's a lot of evidence that the same genes are contributing to all of these outcomes, which we sometimes called externalizing, because they manifest in external ways that we can see: impulsivity, conduct problems, ADHD in kids, substance use problems, etc. And this is really what led me to form, with several other colleagues, what we've called the Externalizing Consortium. We had our first paper come out in Nature Neuroscience a few years back. And what we were able to do is to accumulate much larger sample sizes by looking at all of these different disorders related to this underlying latent disposition toward impulsive action. So how we did this was we went out and we said, what GWAS, what big gene identification studies are already out there for disorders or phenotypes related to impulsivity? And so what was out there, and we arbitrarily said they need to have at least 50,000 individuals, so well-powered GWAS. And so what was out there, it were studies of ADHD, problematic alcohol use, cannabis use, risky sexual behaviors, general risk-taking, and smoking. And what we found is that, in fact, when we looked at, okay, what genes are being identified in all these separate gene identification studies, they also suggested a shared underlying set of genes. And now what we can do is we can do gene identification at the level of this underlying latent vulnerability rather than looking at each disorder by itself. Now what we have is 1.5 million observations, and what we do in our gene identification studies, for those of you that don't eat, sleep, and breathe this like I do, is what we're really doing is we're genotyping markers across the genome. And a marker is just a place where one individual might have one variant and somebody else might have another variant. So somebody might have an A, somebody might have a T, an adenine or a thymine. At that kind of location, what is the variant that's in your DNA. So we are essentially scanning the whole genome, and we're asking for every single one of these positions. We collect a sample of people who are affected or unaffected, or high and low on a particular trait, something like risk-taking, or in some of these studies, affected with alcohol use disorder or unaffected. And we're essentially saying, do people with this particular variant, are they likely to be higher on this outcome or affected with this disorder? So we step through the genome, and this produces something that we call a Manhattan plot, because you ideally want it to have lots of peaks like the skyline of Manhattan. And what it represents is every dot here is one of those millions of tests that has been run for markers across the genome. And the ones that are, these are all the chromosomes and all of the markers tested across the whole genome. And the ones that are peaking up here, that are above this line, are ones that are significantly associated with the outcome after a massive multiple testing correction. And so what you can see is this very much looks like lots of peaks. We identified nearly 600 independent loci, independent locations across the genome that were associated with externalizing, with this broad latent liability toward impulsive action. And so compare that to, at the time, the biggest gene identification studies for alcohol use disorder had identified 29 loci. For cannabis, 2 loci. For opioid use disorder, 1. we identified almost 600. So I think it really powerfully shows how when we take what we have learned about the underlying neuroscience and biology, about the ways that genes are likely to be influencing these outcomes we care about, we can really drastically improve gene identification. And not only that, when we ran the bioinformatics, what we found, no surprise, is that these are genes that are enriched in expression in the brain and in neurodevelopmental processes. So these are genes that are involved in the way our brains are wired toward processing rewards and consequences, and that shows up as more impulsive choices and actions. What we can then do is take all those significantly associated loci and weight each of these variants by its effect size to create something that we call a polygenic score, or a PGS. And what that now represents for any one individual is their overall liability on this dimension toward behavioral under control. And it might manifest as any one of these different types of outcomes. And what we found is that that polygenic score, when we used it to predict externalizing outcomes in independent samples, it accounted for 10% of the variance in independent samples in externalizing outcomes. So that is still to date the most predictive genetic risk score for any outcome. And I'm not going to go into it today, but we actually have in a revise and resubmit at Nature right now an externalizing 2.0 in which we have 4 million individuals in the analysis. We've identified over 1,200 loci. It now predicts upwards of 13% of the variance. And so I think this has been a very successful and exciting way to really move this forward in understanding how genes are influencing risk for alcohol use and problems and other forms of substance use. To give you a sense of now what this kind of prediction in independent samples looks like, what I've done here is we have divided these polygenic scores, which are distributed like a bell curve, back to that, kind of the figure with the mental health jars. The PGS are distributed like a bell curve, and so I have grouped them here just into quintiles. So at the left side, these are individuals with the lowest polygenic scores in the bottom 20%, all the way up to over here, individuals in the top quintile with the highest 20%. And what this is showing is this is actually in a sample that's enriched for having family members affected with substance use disorder. And that's why these rates are particularly high. But it clearly shows that, so these points are indicating what percentage of people here with the lowest genetic risk meet criteria for this upper line for moderate or severe alcohol use disorder. So at the lowest levels of genetic risk, about 30% do, but at the highest levels of genetic risk, about 50% do. Ever using opioids in ways not prescribed by a doctor, that's the bottom line here. At the lowest levels of genetic risk, about 13% of people say they've done this. At the highest levels, about 30%. So clearly there is an association between genotype and outcome, but at the same time, you can be carrying the highest levels of genetic liability on this dimension and not develop substance use problems. I put this slide in here for you all, since many folks on this call study cancer and other related biomedical outcomes. You can't read the details here. If you're interested, the full graph is in our Nature Neuroscience paper. But what this is essentially doing is a FEWAS, meaning we went into the electronic health record data. This is from the Vanderbilt Biobank with our colleague Leah Davis. And we created these polygenic scores for all of the de-identified data in there and said, what is genetic liability toward on this dimension related to in terms of what codes in the electronic health records? And what you can see is that there's many significant findings. In other words, individuals with higher polygenic scores on this externalizing dimension showed worse health in nearly every bodily system. So they were more likely to suffer from heart disease, HIV, diabetes, obesity, lung cancers, liver cancers. In fact, when you start looking at what are the biomedical outcomes that are associated, you will see that many of these are medical outcomes that are mediated by behaviors related to self-regulation. So how much an individual is smoking, drinking, having unsafe sex, eating, etc. And so I think it really speaks to thinking about what are the underlying genetic influences and what are some of these pathways by which individuals might be at elevated risk for some of these downstream medical consequences. Okay, so I have been talking so far about identifying genes involved in this broad externalizing pathway, this kind of broad impulsive action. And that is one way that an individual might carry genetic risk that elevates risk for, say, alcohol problems. But there are also genes that are specific to alcohol problems that are not influencing these other outcomes potentially. And so we're able to then do gene identification at that level and say, okay, what are the genes that just influence risk for alcohol problems, but don't influence these other kinds of impulsive disorders or behaviors? And we've done that. And as you might expect, what you see there are genes that are involved in the metabolism of alcohol. And so what we're really able to start doing is parsing apart the different ways that genes are influencing risk for an outcome. And what we have found is that there are, roughly three big genetically influenced risk pathways. And so there's those physiological genes, those genes that are involved in how our bodies respond to a drug, how much we like it. Does it make us sick or does it make us euphoric? And then there are these externalizing pathways, genes that are involved in how our brains are processing rewards and consequences that show up in a variety of phenotypes related to impulsivity. And I haven't talked about it here today, but we also know that there's a more indirect genetic pathway that's related to how our brains process anxiety and fear. So genes involved in that pathway can indirectly lead to heavier substance use and problems when substances are being used as a way to cope, when there aren't other effective means put in place to address the underlying anxiety or depression. And of course, we are all a combination of lots of these different potentially genetically influenced risk pathways. And so what I'm showing here is in data from one of our samples, the mean number of alcohol symptoms, so higher bars here indicate more alcohol problems, as a function of how many of the high PGS, the upper quintile PGS, an individual is carrying across each of these three dimensions. So you can see individuals who aren't carrying elevated genetic risk on any of these dimensions. They have the lowest levels of alcohol symptoms. If you're carrying that externalizing risk, you have slightly more alcohol problems. Add in externalizing and internalizing, it further goes up. If you have externalizing and the alcohol specific piece, risk further goes up. If you have all three of them, your risk goes up even more. And so the exciting place that I think we find ourselves is when we talk about moving toward precision medicine, we know that our patients represent a heterogeneous cluster of how they ended up with any particular outcome. So they're likely carrying different sets of genetic risk factors, environmental risk factors, etc. But if you look at what I've been talking about today, what we're able to start doing is parsing, at least on the genetic risk part, and I'll get to the environmental side in a minute, into what are these different genetic risk factors, and then how can we prevent and treat based on what types of genetic risk an individual is carrying. And this, of course, all falls under this umbrella that's often called the Precision Medicine Initiative, first announced by Barack Obama at his 2015 presidential State of the Union address, with the idea of moving medicine beyond a one-size-fits-all approach to more personalized treatment and prevention. And I know that in the cancer space, this has really been one of the areas that has been leading the way. When we talk about genetics, I find that people tend to immediately move toward how genetics is going to help on the treatment side of things, that it will help with understanding the underlying biology, and that can help us develop better pharmaceutical treatments, which is absolutely true. And we have work ongoing in that space, which I'm not going to talk further about today since this is more of a prevention group. I'm instead going to talk about how genetics can also be useful on the prevention side. So I'm going to move into this second piece of thinking about how do we translate these genetic findings into something that can meaningfully be used in clinical practice. So when we think about prevention, intervention, treatment for substance use disorders, very often we're thinking about focusing on the environmental pieces, on how do you reduce the risk factors that are found in neighborhoods and communities, in the individual's peer groups, in their settings. And that is crucially important. That's obviously a very important piece of what influences risk. But we're essentially leaving this other really big part of why some individuals are more at risk or what might have led to the development of problems on the table. And so really what we have been working on is how do we also integrate this knowledge of what we have learned about genetic influences on substance use and problems to inform prevention as well. So if we go back to, we've learned something about these different pathways of risk, you'll notice that only one of those, and if you'll recall, it was actually the smallest one, it accounted for the least amount of the heritability, is related to how bodies respond to drugs. The other two genetically influenced risk pathways, especially the big one, externalizing, is not at all related to how bodies respond to drugs. It's related to how brains are wired. And this is a really good news from a prevention perspective, because no one wakes up one morning to discover they've had an onset of alcohol use disorder overnight or opioid use disorder. Instead, we are born with our genetic makeup. And so those genes that have influenced the way our brains are wired toward impulsivity, they show up early on, long before substances usually come into the picture. And so we know that that child who has a brain that is disposed toward more impulsivity is the one that is dangling from treetops, giving parents a heart attack, essentially ending up in the ER more. That same child, if you fast forward a decade or so, we know is more likely to be this person as an adolescent, but not necessarily, because you can channel that same risk-taking disposition into this or into this. And in fact, for full disclosure, that little risk taker was my little risk taker, who in fact comes by it honestly because his father, my brother are in fact all fighter pilots, which is one way to channel a risk-taking disposition. So this brings us to this really important concept that DNA is not destiny. Nobody has a jar that is full from birth. And so knowledge is power. Understanding something about what is in your jar, we can use that to essentially prevent problems before they potentially start. And this is one example taken from many in which we can demonstrate how the environment moderates genetic effects. And so this is a large population-based study of kids over in England. And what I'm showing here is rates of delinquency in adolescence. So all the things that as parents we don't want our kids to be doing, getting into trouble, skipping school, drinking, smoking, etc. This is rates of delinquency as a function of genotype. So those polygenic scores that I talked about earlier for externalizing. And so the yellow line indicates kids in the middle with average genetic risk. The red line is kids at higher than average genetic risk, so a standard deviation or more above the mean on their externalizing polygenic scores. And the blue line represents kids who are carrying lower levels of genetic risk. But it's showing delinquency not only as a function of genotype, but also as a function of parental monitoring. How much are your parents asking about where you're going, who you're with, what you're doing? And what you can see is that parental monitoring is good for all kids. The rates of delinquency go down as parents are more actively engaged in their kids' lives. But it goes down the fastest for the kids who have the highest levels of genetic risk. So at lower levels of parental monitoring, when kids have more free reign, more opportunity to express their genetic predispositions, you see the expected relationship between more genetic risk, higher rates of getting into trouble and delinquent behavior. But at higher levels of parental monitoring, there's no difference in the rates at which kids are engaging in delinquent behavior as a function of genotype. So, what we've been working on is how do we take all this knowledge and translate this now into something that could be clinically meaningful and useful in a prevention setting? So, we know a lot about what are those marbles, right? What are the different genes and how they work? And we also know a lot about what are the environments and the early behaviors that indicate elevated risk. And we know this from longitudinal developmental studies where we've followed kids from childhood into adulthood and looked at what things are most predictive of who goes on to develop problems. And several of those are listed here. They're traumatic life events. They're having a family history. They're having substance using peers who are providing access and acceptance. It's early initiation of use. It's other kinds of challenging life situations. These are all environmental and behavioral factors that can also elevate risk. And it turns out that when you combine all of these pieces, the genetics with behavioral and environmental risk factors, we can powerfully differentiate how at risk someone is of developing a substance use problem. And this, I think, creates this exciting idea that we can potentially prevent problems before they start. And so, what this little graphic over here shows is that individuals who are carrying in these big longitudinal epidemiological samples the highest levels of genetic, behavioral, and environmental risk, 84% of them went on to develop a substance use disorder. Individuals who are carrying the lowest levels of genetic, environmental, behavioral risk, only 4% of them did. We know a lot about how at risk an individual is based on things that are happening earlier in life before substance use problems start. And so, what we have done is we have worked to develop algorithms and an online platform. If you're interested, you can scan this QR code and it will take you to the website where you can learn more and kind of see the program. We call it CARES, the Comprehensive Addiction Risk Evaluation System. And what we're trying to do is take these decades of research and millions of dollars that have gone into identifying all these risk factors and put them together in a way that we can essentially help people understand their risk. And so the way that the platform works is an individual creates an account, they answer a short survey which contains those behaviors and environments that are most predictive of who developed problems. We send a kit home and an individual can do a buccal swab or saliva tube and send it back to the lab. We essentially scan the genome, we create these polygenic scores, we put all the information together, and we partnered with psychiatric genetic counselors to essentially create language that was empowering, non-stigmatizing, non-determinative to help individuals understand their risk profile. Which of these factors, genetic and behavioral, environmental, which of these risk factors might they be carrying? How does that translate into their overall risk? But importantly, we don't just want to tell someone they're at risk. We then want to connect them with evidence-based resources to reduce risk. And we tailor those resources to the types of risk that they're carrying. Are they carrying more of this externalizing, more of this behavioral under control risk? Are they carrying more of the internalizing risk, etc.? And excitingly, we just hot off the press, we are in the process of writing it up right now, but we ran an RCT, we enrolled young adults, 400 of them, and what we found is that individuals who got, and we tracked essentially their substance use then across time, 30 days and 3 months later, and we found that in fact, individuals who were getting feedback that they were at higher risk significantly reduced their alcohol and other drug use across that period of time. So we think this is very encouraging. And this is, in fact, then what led to my spinning off this startup. I partnered with someone who lost his father to substance use problems, and he found me kind of through a circuitous route, which I'd be happy to talk more about. It's a story that involves Slash and the music world and all kinds of crazy things, and essentially said, we need to do more to get this research out to people. And so that's kind of how that came about. I'm going to switch gears and just very briefly talk about one other area where we're trying to use genetics for prevention. And this is related to, we are here at a university. Universities are places where we have college students that are engaging in high rates of risky substance use. We also know that they are highly unmotivated to actively participate in anything that sounds like substance use prevention programming. And at universities, we have all kinds of ways of forcing them to participate in some of our substance use programming. Often we'll make them go through an online program before they can register for their classes. We have been trying to find, and this is work I started with colleagues at VCU and have continued here at Rutgers, creative solutions to essentially try and bring these new research advances and this information to young people in ways that will essentially connect more with them. Or it's what the mom and me calls chocolate covered broccoli. How do you get something that's good for someone to them in a way that they are also excited about? So it turns out college kids don't want anything to do with something that sounds like adults who are coming in doing substance use prevention programming. But what they love to do is fill out questionnaires about themselves. So we thought we're going to capitalize on that. We're going to capitalize on this kind of gamification and things that you see in social media. Let's try and use it for good. And we created a program. We do not sell it as substance use prevention. We call it All About You. Learn about yourself to get the most out of your college experience. What they do is they fill out a series of questions. And of course, what those questions are is they are tapping into those externalizing and internalizing dimensions that we know elevate risk for substance use problems. So we don't even have to, get buccal swabs or DNA. We can provide feedback on those dimensions. And we talk about the good, the not so good associated with it, and level up, kind of ways that essentially one can take that information and channel it to be your secret superpower and not have it get you into problematic situations. What we found in the initial RCT is that students who went through this program were less likely to use alcohol and cannabis compared to students who went through the gold standard harm reduction programs that most universities continue to use today. And so very excitingly, Rutgers actually rolled this out just this summer with all of its 9,000 incoming freshmen. So this will be the program that our students are going through, which will also allow us then to have that data collected in a de-identified way to connect back to the research enterprise to really essentially engage students in research, to invite them to be a part of the research process so we can really start to link research with our prevention programming and our academic mission. Okay, there we go. Now it's advancing. So to wrap up, why is this all so important? Coming back to the beginning, I think that we can use basic science advances to really help change the addiction prevention landscape. And that understanding how genetic influences shape risk for substance use problems and these related downstream medical disorders really raises new possibilities for personalized prevention, intervention, and treatment. I want to end by acknowledging that everything I have presented today has been, science is a team sport, has been the product of many, many individuals who have contributed to these large data sets that we have analyzed, to a lot of funding that we have been fortunate to receive from the National Institutes of Health, to the entire team of investigators that are involved in these large international collaborations and that share data so freely to make all of these scientific advances possible. As I mentioned at the very beginning, we have a really vibrant addiction community here at Rutgers. I have been very privileged to join it and to have the honor of getting to help shepherd and lead this community. If you want to learn more about it and ways that you can get involved, you can go to addiction.rutgers.edu or scan this QR code. And I thank you for your attention, and I am happy to take any questions that you all might have.
Thank you so much, Dr. Dick. That was amazing, very exciting and cool. Just what some of us needed today. Uh, so if people want to turn on your cameras, we have a few minutes for questions. I see there are a couple already in the chat. Dr. Weiss was asking about honing in on rare alleles and how does that fit into the polygenic risk score analyses.
Oh, hi, Stan. Thank you for that question. So you are correct that the polygenic scores that I talked about are largely capturing common variation. In substance use disorders, that's been most of what we have focused on, although we actually have a center that is under review at NIDA right now, or we'll be going back in. And we're partnering with Zishan Ahmad and the AI and Data Analytics Core at the Institute for Health. He has been actually applying machine learning and other kinds of bioinformatics techniques to a pilot sample of 23 individuals we collected through our treatment settings. And actually, what he did find is a lot of personal mutations. So, in other words, rare variants that did appear to be playing a role in this admittedly just initial pilot sample. And so, what we're really hoping is this center grant that we've put in would essentially partner with our clinical treatment settings to enroll individuals in substance use disorder treatment programs and then to do extensive phenotyping. So, multi-omics, deep phenotyping, imaging, and EMA data collection. And so what we would then essentially be doing is integrating through AI techniques all these different pieces to identify subgroups and are there different profiles of risk and hopefully identifying biomarkers. And but it's really interesting that in these first individuals, it does look like there's some rare variation that is showing up that we have not captured in these previous studies. So the hope is to bring these pieces together eventually. Thanks for that question.
Yes, and then Sharon was commenting on the parental questions and having school programs.
So, yes, the part of the reason that I briefly wanted to mention the All About You program that we've really worked to build is that it is a way to get at, genetic risk and behavioral and environmental risk, but in ways that doesn't require genotyping or many things that start to raise ethical questions and whatnot, especially with minors. To do, an online prevention program where students are learning about themselves and how to use that to make the best choices for themselves as they're moving through adolescence into young adulthood is something that we think very well fits within school prevention programs. We actually have a grant underway to partner with schools across New Jersey. We have created through a philanthropic gift that we've received at Rutgers. We are taking the dashboard that we built for all the Rutgers students and creating a generic version where schools will be able to put in their own mascots, their own colors, their own language. So the science will remain the same, but then they can also fill in their own resources that are available at that school. So there'll be kind of a default to broadly of it, national resources and other sorts of things that are tailored to different risk profiles, but then schools can either add or substitute their own. So we are building out that dashboard right now to be able to make it available and then hopefully to partner with schools to see how well it works. And of course, the hope would be to engage parents in that process as well, too. And so we have some of these initiatives that either, are student focused or that also bring in the parents because I think this is important information for parents to understand too. I give a lot of parent talks and they always say, I wish I could know if my kid is the one who's going to be at risk. And I would say, tell me about your kid. I can tell you, how much risk they're carrying in a sense. So we are trying some of these creative ways to get this information out. The book that I had on my last slide there is actually a parenting book. I showed my little risk taker earlier on. Um, Oh, the irony. I found myself raising the high-risk child that I study, highly impulsive, highly emotional, and I realized how much of this research was not getting out to parents and how they were blaming themselves, questioning themselves, wondering what's wrong with their child when they were more impulsive or more anxious and so I really wrote the book. It's not a research book. It is me as a parent who happens to be a researcher for other parents to try and bring the research to them. So I went off on a tangent there, but these are some of the many ways that we're trying to reach different constituencies, parents, kids, in addition through our hospital setting where we are piloting cares as well.
I was thinking more about parents asking questions. I mean, that used to be one of the big campaigns, I think, when back in the day about smoking, right? Just asking questions about the child's whereabouts, history of divorce in the family, a single parent. Those are at-risk factors that would make something the parent could ostensibly at least do something like asking more questions and how to frame those questions for a preteen or teen, those at-risk years. I just thought more like identifying parents who might be able to at least understand what risk their child's at and then how to talk to them.
Yes, absolutely. And certainly there are other colleagues, both at the RARC and beyond, who are focused more on the
Yeah.
how do we identify those parents, support those parents. I 100% agree that's really important. Like I said, I tend to do a lot of parent talks because as I had a child going through the school system, parents can play such an important role. And one of the things I usually always put up is that NIDA has resources, as I'm sure many of you know, about how to talk to your kids and those kinds of things. So yes, I agree that's critically important. And we're always trying to think of more ways to get the message out. Oh, I'll mention briefly, we are actually going to just this fall formally announce, so don't tell anybody yet, but you're getting a preview that we are partnering with Partnership to End Addiction. The CEO, which happens to be based in New Jersey, and the CEO is a Rutgers alum, and they are the largest nonprofit dedicated toward working with parents, caregivers, etc., on how to prevent substance use in kids, uptake in kids who have problems. And so we are going to be, kind of formally partnering with them as another pathway by which to take research, to try and get it out to parents and other relevant caregivers.
So cool. And I see Dan Rosenblum was asking about ancestry, genetic risk based on ancestry, and how that might play out in research.
Yes. So, Dan, thank you for asking that important question. You're someone who I know knows all the details of how these things work. And of course, so these polygenic scores, they are most predictive for individuals whose genomic backgrounds are most similar to the individuals who are in the discovery sample. And of course, that is because of the ancestral migration of peoples, around the world over the course of history, such that people who are from similar parts of the world have a more similar genetic background. And as we're identifying these genes, sometimes we're really close to them. And so when you're on a genetic background that's more similar, then it's more likely to pick up those risk variants in individuals from that same, roughly ancestral group or genomic similarity group, if you will. The polygenic scores on that first wave of externalizing were from a sample that was entirely individuals of European genomic descent. And this is a huge problem, as many of you may know, across all areas of genomics. There are far more genetic samples available for individuals of European genomic background than other areas of the world. We are working to change that. It's going to take time to amass more samples and to genotype them. And I am very happy to say in externalizing 2.0, which I alluded to, we have been able to incorporate more diversity. So we're able to incorporate genomes that are of African ancestral descent as well, too. That was the only other group for which there were big enough samples, though, which is a problem. We have a grant underway to collect more samples of Asian genomic background, East Asian. So we're trying to do better on this. We're both developing methods to make those PGS predict better into other groups, and we're trying to amass more samples. The other piece is because we're not using the genomic scores only to predict outcomes, we are also using the behavioral and environmental risk information. Because many of those behaviors represent and are capturing genetic risk, right? So impulsivity is capturing a certain degree of genetic risk. And so that is still picking up on some of that genetic risk, even for individuals whose backgrounds are not as closely matched and the polygenic scores are not as accurate.
Well, I'm sure we would want to listen to you for a few more hours, but you may have to go and do something else, Dr. Dick. So thank you so much. Very inspiring. And thanks
Well,
again.
thank you for
you so
having
much.
me. Let me know if there's ever ways that I can further support, connections and collaborations.
Yes, we'd love to.
Mhm.
Bye.
Thank
Take
you.
care.
Great talk.